In the 1950s, Francis Crick proposed his adapter hypothesis to explain how gene expression occurs in DNA via protein synthesis. This meant that RNA molecules act as the adapters that recognize the amino acids once they receive them. From there, the RNA works with the messenger RNA, also known as mRNA, for base pairing, and the adapters add to the polypeptide chain. When the sisters need to make a brew in the cauldron, what they are really doing is translating and transcribing DNA from their book, with DNA being the brew. The cauldron is the mRNA, which needs interaction from the codon base pairing in order to work. Mary is the codon on the end of the mRNA, as she always pairs with her sister Sarah, the anticodon on the end of the tRNA, to let the magic happen. Winnie is the adapter between these two, the tRNA, who is the star witch of these brews, and adds her sister's input to them, like a tRNA adds amino acids to the polypeptide chain. The polypeptide chain here is their magic when they all link together and cause protein synthesis and ultimately more DNA. So where did Francis Crick get this idea from in the first place? We know that transfer RNA molecules are the specific RNA that act as the adapters, but further, Crick was inspired by the central dogma. The central dogma uses transcription and translation to turn DNA into proteins. First, DNA is transcribed into mRNA. From there, the mRNA is translated into the sequence of amino acids known as the polypeptide chain. Crick suggested that there would be 20 different adapters for each amino acid, and we see this through the side chain illustration and chart. These 20 amino acid side chains and their relationship to the codons are what make up the universal genetic code. He knew his hypothesis to be true because mRNA has nucleic acids made from nucleotides that only recognize nucleic, not amino acids, meaning there must be a need for the presence of an adapter such as tRNA. The tRNA acts as the bridge between mRNA codon and the amino acids, and without it, something would be missing. Here's a diagram of how tRNA identifies the codons on mRNA via base pairing. As we learned through Hocus Pocus, codons and anticodons are complementary. The amino acids bind on the 3' end of the tRNA and the anticodon on the 5' end. Lastly, for the central dogma, let's talk about the ribosomes. I mentioned earlier that the central dogma uses transcription and translation. Ribosomes are where translation happens, and they are found at the 5' end of mRNA. If an alteration occurred that prevents a tRNA from acting as an adapter that follows a universal genetic code, there would be a mutation in the DNA. In Hocus Pocus terms, we have gone off book and messed up our spell. This mutation may cause the tRNA to charge with the wrong amino acid because of aminoacyl enzyme, or possibly the anticodon on tRNA will not bind with its codon and begin translation in the first place. Either way, the amino acids will not be read as genetic information code, and the protein will not be synthesized into DNA because the cell is not growing. Thanks for watching!